Hello students, in today's module we will learn about red blood cell membrane. Modern knowledge of the red blood cell or the erythrocyte plasma membrane and its membrane skeleton began with Marchesi and Steer's identification of spectrin in 1968. Before that, almost the only thing known about membranes was that they contained a lipid bilayer. The red cell membrane constitutes only about 1% of the dry weight of the erythrocyte. Nevertheless, it is an important organelle that serves as a boundary with a surface area of about 140 micrometer square, thus determining the bioconcave shape of the erythrocyte. It provides the red blood cell with a deformable and resilient surface that enables it to traverse capillary and splenic channels smaller than half its diameter. Because of its unique transport functions and its selective permeability to cations, erythrocyte membrane regulates the contents of the red blood cell and maintains an ionic gradient between the intracellular and the extracellular environments. Finally, it regulates the interaction of the erythrocyte with neighboring cells and with the surrounding medium. More is known about the plasma membrane of the human RBC than about any other eukaryotic membrane. There are a number of reasons for this. Red blood cells are available in large numbers relatively uncontaminated by other cell types. Since they have no nucleus or internal organelles, the plasma membrane is their only membrane, and it can be isolated without contamination by internal membranes. It is easy to prepare empty red blood cell membranes, or we call ghosts, by putting the cells in a medium with a lower salt concentration than the cell interior. Water then flows into the red cells, causing them to swell and burst and release their hemoglobin, which is the major non-membrane protein. Membrane ghosts can be studied while they are still leaky, or they can be allowed to reseal so that water-soluble reagent cannot reach the internal phase. Moreover, sealed inside-out vesicles can also be prepared from red blood cell ghosts, and so the external side and the internal cytoplasmic side of the membrane can be studied separately. The use of sealed and unsealed red cell ghosts led to the first demonstration that some membrane proteins extend across the lipid bilayer and that the lipid compositions of the two halves of the bilayer are different. Biochemically, the red cell membrane is composed of proteins, lipids, and carbohydrates. The carbohydrate moiety is associated either with membrane lipids, we call them glycolipids, or with membrane proteins as glycoproteins. Now let's study the red blood cell membrane lipids. The erythrocyte cell membrane comprises a typical lipid bilayer similar to what can be found in virtually all human cells. Phospholipids, cholesterol, and glycosphingolipids are the three major constituents of the erythrocyte lipid bilayer. About 50% of the fatty acids in red blood cell membrane, phospholipids, are saturated and 50% are unsaturated. The lipid composition is important to many physical properties like membrane permeability and fluidity. Also, the activity of many membrane proteins is regulated by their interactions with lipids in the bilayer. Unlike cholesterol, which is evenly distributed between the inner and the outer leaflets, the 5 major phospholipids are asymmetrically distributed. The outer monolayer is comprised mostly of phosphatidylcholine and sphingomyelin, and the inner monolayer mostly of phosphatidylethanolamine, phosphatidylinositol, and phosphatidylserine. The maintenance of an asymmetric phospholipid distribution in the bilayer is critical for cell integrity and function. Glycosphingolipids account for about 3% of the molar concentration of membrane lipids. Now let's study the red blood cell membrane proteins. When the plasma membrane proteins of the human red blood cell are studied by SDS polyacrylamide gel electrophoresis, approximately 15 major protein bands are detected, varying in molecular weight from 15,000 to 150,000. 3 of these proteins, spectrin, glycophorin, and band 3, account for more than 60% by weight of the total membrane protein. Each of these proteins is arranged in the membrane in a different manner. We can use them as examples of 3 major ways that proteins are associated with membranes, not only in the red blood cell, but in other cells as well. Structural or supporting proteins. At least 5 or 6 major polypeptides of the human erythrocyte support and stabilize the membrane. Bands 1 and 2, which is spectrin, and 5, which is actin, join together to form a submembranous cytoskeleton of the red blood cell. Band 2.1, which is encheirin, 2.2 to 2.6, which is cyndines, and 4.1 to 4.2 have been implicated in linking spectrin to the membrane. The viscoelastic spectrin actin microfibrillar meshwork influences cell shape and provides anchoring sites at the cytoplasmic membrane surface for transmembranous proteins, thus limiting their lateral surface mobility. Most of the protein molecules associated with the human red blood cell membrane are peripheral membrane proteins associated with the cytoplasmic side of the lipid bilayer. The most abundant of these proteins is spectrin, which is a long, thin, flexible rod about 100 nanometer in length that constitutes about 25% of the membrane-associated protein mass. It is the principal component of the protein meshwork, which is the cytoskeleton that underlies the red blood cell membrane, maintaining the structural integrity and biconcave shape of the membrane. If the cytoskeleton is dissociated from the red blood cell ghosts in low ionic strength solutions, the membrane fragments into small vesicles. Spectrin is a heterodimer formed from 2 large, structurally similar subunits. The heterodimers self-associate head-to-head to form 200 nanometer long tetramers. The tail ends of the 4 or 5 tetramers are linked together by binding to short actin filaments and to other cytoskeletal proteins, including the band 4.1 protein, in what we call a junctional complex. The final result is a deformable, net-like meshwork that underlies the entire cytoplasmic surface of the membrane. It is this spectrin-based cytoskeleton that enables the red cell to withstand the stress on its membrane as it is forced through narrow capillaries. Mice and humans with genetic abnormalities of spectrin are anemic and have red blood cells that are spherical instead of concave and abnormally fragile. The severity of the anemia increases with the degree of the spectrin deficiency. Binding of the spectrin depends on a large intracellular attachment protein called encheirin, which attaches both to spectrin and to the cytoplasmic domain of the transmembrane protein band 3. By connecting some of the band 3 molecules to spectrin, encheirin links to the spectrin network to the membrane. It also greatly reduces the rate of diffusion of these band 3 molecules in the lipid bilayer. The spectrin-based cytoskeleton is also attached to the membrane by a second mechanism, which depends on the band 4.1 protein mentioned before. This protein, which binds to spectrin and actin, also binds to the cytoplasmic domain of both band 3 and glycophorin, the other major transmembrane protein in the red blood cells. An analogous but much more elaborate and complicated cytoskeletal network exists beneath the plasma membrane of nucleated cells. This network, which constitutes a cortical region or the cortex of the cytoplasm, is rich in actin filaments, which are thought to be attached to the plasma membrane in numerous ways. Proteins that are structurally homologous to spectrin and chirin and band 4.1 are present in the cortex of nucleated cells, but their organization and functions are less well understood than they are in red blood cells. Catalytic proteins. The anion transporter, or band 3, is the predominant intrinsic membrane protein, comprising approximately 25% of the total membrane protein of the human erythrocyte. Band 3 is a glycoprotein that contains 5 to 8% carbohydrate on a weight basis. It derives its name from its position relative to the other membrane proteins after electrophoresis in SDS polyacrylamide gels. Band 3 is a multi-pass transmembrane protein, traversing the membrane in a highly folded conformation. Each red blood cell contains about 106 band 3 polypeptide chains, which are arranged as dimers in the membrane. The main function of a red blood cell is to carry oxygen from the lungs to the tissues and to help carry carbon dioxide from the tissues to the lungs. The band 3 protein is crucial for the second part of these functions. Carbon dioxide is only sparingly soluble in water, and so it is carried in the blood plasma as bicarbonate, which is formed and broken down inside red blood cells by an enzyme that catalyzes the reaction of water and carbon dioxide to bicarbonate and hydrogen ions. The band 3 protein acts as an anion transporter, which allows the bicarbonate to cross the membrane in exchange for chloride ions. By making the red blood cell membrane freely permeable to bicarbonate, this transporter increases the amount of carbon dioxide that the blood can deliver to the lungs. Another important catalytic protein is a sodium-potassium ATPase complex responsible for the active transport of sodium and potassium ions across cell membranes. It is an integral protein that spans the red blood cell membrane and contains both a large polypeptide of approximately 95,000 daltons and a smaller glycoprotein of about 45,000 daltons. Besides the catalytic proteins we just mentioned, there are at least 35 to 40 enzymes, including glucose transport proteins, whose activity is confined to the membrane. So most of the enzymes of carbohydrate metabolism and the enzyme acetylcholine esterase are externally oriented on the surface of the red blood cell. The enzymes calcium ATPase, like sodium potassium ATPase, are integral proteins that span the red blood cell membrane. Contact or receptor proteins. Receptor or contact proteins are those components on the surface of red blood cell that mediate its interaction with the surrounding environment. They have unique molecular features that confer biologic specificity and conformation. Biochemically, they are either glycolipids or glycoproteins in nature. The sialoglycoprotein, glycophorin A, is a sialic acid-rich glycoprotein and to date is the best studied contact protein and is the prototype of receptor glycopolypeptides of the erythrocyte. Like a typical transmembrane protein, the major sialoglycophorin of the human red blood cell, glycophorin A, is a small single-pass transmembrane glycoprotein with most of its mass on the external surface of the membrane, where its hydrophilic amino terminal end is located. This part of the protein carries all of the carbohydrate, which accounts for 60% of the molecule's mass. In fact, the great majority of the total red blood cell surface carbohydrate, including more than 90% of the sialic acid, and therefore most of the negative charge of the surface, is carried by the glycophorin molecules. The hydrophilic carboxyl terminal tail of the glycophorin is exposed to the cytosol, while a hydrophobic alpha helical segment spans the lipid bilayer. Since glycophorins are rich in sialic acid, it gives the RBCs a very hydrophilic charged coat. This allows them to circulate freely without adhering to other cells or to the walls of blood vessels. Now let's look at the blood group antigens. Of all the blood group antigens, only the MN determinants are definitely localized to glycophorin A. The I antigen and some Duffy blood groups may be localized in the extracellular domain of band 3. Erythrocytes that lack the Duffy blood group antigens are resistant to invasion by Plasmodium vivax. The ABH and P antigens are both glycosphingolipids in nature, but their relation to the lipid bilayer and other surface proteins is not well established. The Rh determinants are lipoproteins, and their absence in the Rh null syndrome renders the erythrocytes stomatocytic in shape with decreased lifespan and increased potassium transport across the membrane. The mechelioid phenotype, a Kell null variant, is accompanied by abundant acanthocytes, reticulocytosis, and a compensated hemolytic state. Localization of the blood group antigens on the red cell surface and its implication in the structure-function relationship is a fascinating field. To conclude, the red cell membrane skeleton is a pseudo-hexagonal meshwork of spectrin, actin, protein 4.1 R, and chirin and actin-associated proteins that laminates the inner membrane surface and attaches to the overlying lipid bilayer via band 3 containing multi-protein complexes at the ankyrin and actin binding ends of spectrin. The membrane skeleton strengthens the lipid bilayer and endows the membrane with the durability and flexibility to survive in the circulation. A great deal has been learned about the structure of the red cell membrane and the membrane skeleton since the first model was published. Because the red cell membrane skeleton is the paradigm for studies of spectrin-based membrane skeletons in all cells, and because we now know that spectrin and other membrane skeletal proteins invest internal organelles, some transport vesicles and plasma membranes, and even play a role in nuclear organization, it is more important than ever to understand the structure and function of the red cell membrane skeleton. Thank you.